Hello everyone, my name is Eva Collakova. I'm a researcher at Virginia Tech, and this is my best friend here. Well, not really best friend, but this little plant is called Arabidopsis thaliana, and a lot of scientists like myself use it as a model system because it has a lot of advantages. As you can see, it's quite small. And it can produce quite a lot of seeds, grows very fast, and the genome has been already sequenced. And there's all these tools that are out there that we can use to study this plant. And because of that, we use it as a model system rather than working with big crops like soybeans or canola, which take more time to grow, and we don't know as much about them. So, do you guys know why the seeds produce oils, sugars, and proteins? Anyone? Well, let's think about it. When you plant a seed in the spring in your garden, it needs to germinate before it becomes a plant. And in order to do that, it needs to have food and energy. And if you look at the seeds, they're not green, so they're not able to produce their own food and energy. And that's what these storage compounds are for. They provide food and energy for germinating seeds. Also, oils, proteins, and sugars are an important source of food for humans and animals. And in recent years, the oils have been used as biofuel. Now, how can we make more biofuel in these seeds? Well, in order to do so, we need to understand how these compounds are formed. Then we can genetically engineer the seeds to produce more oil that can be used for biofuel. Alright, so we use modern biotechnology tools to find genes that are involved in controlling oil accumulation in seeds, and also to genetically manipulate these genes to make more oil. We use a number of different machines for different tasks, but it's not just the machines that we use as tools. Plants with different sets of specific genes can be tools. For example, plants that are mutants, they have specific genes inactivated or knocked out. Using mutants allows us to identify these genes because we can ask a question. Do we see any unusual characteristics, or as we call it, phenotypes, when this gene is inactive? Well, in our case, we're looking for changes in seed composition. And by that, I mean specifically whether mutant seeds contain more or less oil than the normal plant, and we call this normal plant wild type. Alright guys, so now we're ready to analyze the seeds and see if there's any changes in oil levels. And this is the machine that we do it with, and it's called gas chromatography mass spectrometry, or simply as we call it, GCMS. And so we can load our samples here in the tray, and then the robotic arm will bring it into this tower where the sample is injected in the machine and analyzed. Alright, so we have actually identified several genes that affect oil levels in Arabidopsis seeds, and this fall you'll be working with some of the mutants that show changes in seed composition. And this is actually where I will need your help. So after you're done with your treatments, what I want you guys to do is collect some seeds for me so I can analyze them on this machine to find out how your treatments affected oil levels in the mutant versus wild type. And then we will share the data, and hopefully together we can discover how these treatments affected seed composition and contribute to scientific knowledge together.